Okay, my friends, hold on. This is going to be interesting. We all know that there's membranes. We all have skin, and that protects you from invasion. Not only does the skin protect you from invasion, but every single thing in your body has a membrane, and that membrane protects that particular thing, whether it's an organ or a muscle or a tendon or a bone or whatever it is. It has a membrane, and there's hundreds and hundreds of different types of membranes, and they all have their own chemistry. And this is where we're going right now, as to what is the chemistry and what is the cellular makeup of the things that are in that chemistry, because they contain enzymes. Now, this is strictly research. It's things that I believe have been missed, and we're going to go through them right now. And only research. There's no medical advice given here whatsoever. You have to start with research. All right, let's just start off with the basics. Yes, the human body is made of trillions of cells. Everything's a cell. All your skin's cells. Your eyeballs have cells. Everything that's in you, your blood cells are cells. Everything that is in you is made in a cellular compartment. And each one of those fundamental units of human life, some of them work to absorb oxygen. Some of them work in your lymph nodes to get rid of toxins. Some of them do other things, your kidneys, your urine, your digestion, everything. 100% is controlled by cells. What controls cells? That's the key. This is what controls cells, is the cell membrane. All right? It's a plasma membrane, and it separates the cells inside the interior from the fluids that are surrounded, the exterior environment. The cell membrane is semi-permeable. It allows some stuff in, but not everything. Well, what controls what it allows in and what it doesn't allow in? It regulates what materials can enter. How does it regulate it? And what can exit the cell? How does it do that? As well as how much of a substance can enter. It's made up of a bilipid layer of phospholipids forming a stable barrier around it. But it has little tiny entryways and so forth. You'll see in a minute. And it's separated from these two aqueous solutions by this membrane. It's in the middle. Okay, we know we're made of trillions of cells. Well, are they all exactly the same? Maybe a little different color or something? Well, no. There's specialized cells, neurons, that do transfer information, and people get, conditions, neurological conditions, because they've got something wrong in that area. Muscles, totally different. Spermatozoa, totally different. Stem cells, totally different. Egg cells, skin cells, all kinds of different cells. And every single one of them is a different chemistry inside the envelope. All right, so don't forget, trillions of cells and all kinds of different muscles and tendons and eyeballs and, membrane, all kinds of things. So let's take a look at what a cell looks like. Okay, this is what they say from the National Institute of Health, and this has just been revised, within the last 5 or 6 years. Thoroughly revised estimates show that the typical adult human body consists of about 30 trillion human cells and about 38 trillion bacteria. So there's more of them than there are of us in us. And this is the National Institute of Health, so this is not just nobody. Okay, I mean, I got to be honest with you. I find this fascinating. Can you imagine there's trillions of these, and many trillions, and there's many trillions bacteria. They all have a membrane on the outside. That's the membrane. All these little holes are they call them ion channels. They're channels that allow things to enter, but only things that they want to allow to enter, and only things they want to allow to exit. That's it. So there's some very intelligent situation going on here. How does it know what it wants to allow in, and how does it know what it wants to expel? Nobody knows. Now, these are the things I want you to concentrate on. Ribosomes. All right, ribosomes. Very, 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 1,000 times very important. A ribosome is the product of a bacteria. And the ribosome creates an enzyme, and the enzyme is the thing that does literally all the work. And I'll show you this, and if you don't have the right bacteria to create that ribosome, you're not going to get the chemistry. We're going to go into the chemistry shortly. These are all the different things in here. You don't have to know much about it other than that this is the cell membrane. These are the inlet pores, and the excrement can go out through them. All these are different, what they call little organelles, mitochondria, and then you have a whole nother central core, which is called the nucleus. All right, and then the nucleus has its own nuclear envelope, which is another membrane. These all have membranes. These have membranes, and membranes everywhere. There's membranes everywhere. Now, the chemistry of this is 100% different. The chemistry of that, and the chemistry of that, and the chemistry of this, and the chemistry of that, and that, and every one of them has different chemistry. How does the chemistry become created? How do you know how to make this chemistry? Enzymes make the chemistry. I'll show you right now. Now here's the answer to the question. Does bacteria create ribosomes? Yes. Bacteria create ribosomes in their cytoplasm, which we talked about a second ago. And they assemble these ribosomes. Now let me show you what the ribosomes do. Well, ribosomes are enzymes. Enzymes can speed up chemical reactions by millions to trillions of times. For example, carbonic anhydrase can catalyze over 1 million reactions per second, which can make a reaction billions of times faster than normal. They can do, within 1/4 of a second, more chemistry than that would happen in years if you didn't have the catalyst. And the catalyst is nothing more than a huge molecule like this that has a zillion different magnetic moments. And I'm going to show you this. I'm going to show you everything that I'm talking about. And I'm not making any guesses here. They know what they look like. And they know they don't get used up. They come along and click things, click, 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 and kill them, break them down, whatever they do to them. And it's an electric discharge from this to them. And these don't even get used up. Eventually they die off, or something happens to them, because they have to get out of your body somewhere. But they do as much chemistry as necessary, and then apparently they just go into the system and get flushed out. But this is how fast they can speed up reactions, and I mean it's just unbelievably fast. All right, so don't forget, yes, bacteria create the ribosomes. All right, this is just beyond outrageous. What I want you to focus in on is ribosomes, all right, and lysosomes. Well, what are they? All these other things are organelles as well, and they are organelles. Well, what does a ribosome do? And what does a lysosome do? Because those are the most important ones that we're going to talk about. The rest have specific functions. Mitochondria gives you energy and so forth. But these two are the ones. Ribosomes create the enzymes, and lysosomes are the slicers and dicers. All right, this is all I want you to concentrate on. Don't forget, there's 38 trillion different bacteria in us, and of that, there's probably hundreds of thousands of species, or I don't know how many. That's just a total guess. I have no idea, but there is a ton of them. Now, every one of them has its own DNA, and that DNA programs this bacteria to create this ribosome. That's what it does. Now, not every bacteria, I don't think, create ribosomes. Some of them are consuming bacteria. They go out and they consume waste and so forth. And they have flagellas on them and all these little fibers and all that stuff. But they also have a membrane, plasma membrane, just like all of them. But this is what I concentrate on right here, the ribosomes. That's what squirts out and creates the enzymes. Without the bacteria, you don't get them. All right, that's an enzyme. That didn't come from nowhere, and that does an exact specific job. They have to be exactly like that, or it doesn't work. Whatever the program is, to program this to have all these different little magnetic moments. And what happens is they have what I call click chemistry. They come up against other molecules and they go click, and then a whole batch of them die at once, instantly. And that's why they speed things up trillions of times, it says, up to trillions of times. And it's done. But the only way you get this is from a ribosome. And the ribosome unfolds to create these enzymes. And also they can be proteins. Proteins build things, enzymes break things. All right, here's what's going on. This is the ribosomes, and they're there for a purpose made by a certain bacteria to do an exact certain job, whether it's to break down carbohydrates or fats or to attack certain invading other molecules, whatever it is. And these are programmed, literally programmed. It's a program. I don't care how you look at it. It is a program. I'm going to show you it's a program. And when this thing senses that it's in the vicinity of something it wants to kill, it unsheaths. So in other words, it's a ball like this. It comes down and goes, and then it comes out like this. And then it goes and attacks everything with an enzyme. Or it comes over here and makes up a piece of bone or something, a protein. So all it is a program to create chemistry to destroy or break things down or to make things up. All right, very, very simple. Single cell, that's a single cell, tiniest thing there is. And it squirts out these little tiny balls that are called ribosomes. This whole thing is a ribosome. And the ball comes out, and inside of that ball is this chain of particles. And they start to come around like this, and they come down and they program using an enzyme. Anytime you see a cloud like that, it means there's an enzyme in there. I believe it's transferase. And the transferase transfers this into the messenger RNA. So now you have a single strand of messenger RNA programmed to whatever this ribosome was programmed by this bacteria. You don't have the bacteria, you don't get the program. This is not going to happen accidentally. Now, out comes the messenger RNA, and what does it do? It has to find a way to get into the DNA. This is our DNA. And if it can get in here, and there's a way it can, and I'll talk about that in a minute, the RNA polymerase, which is another enzyme, breaks everything down. So, all right, get in there, and it comes out. This here goes program, and this one, the polymerase gets everything out of the way so it can get inside. You have to have these enzymes. You don't have these cloudy little enzymes, you're done. All right, this is important. Lysosomes are organelles found in the cells, act as cells' digestive systems, breaking down materials from inside and outside the cells. They contain about 50 different enzymes. That means they have to be 50 different ribosomes because they create the enzymes. And they break down biological polymers such as proteins, nucleic acids, carbohydrates, lipids, fats. The enzymes are called acid hydrolases and are active in acid environment. A pH of around 4.8 inside the lysosome compared to 7.2 in the cytosol, which is the fluids outside. Now, let's take a look at what that lysosome looks like. And don't forget, 50 different enzymes just in the lysosome. All right, and I have no way to verify this one way or the other, but the way they're making it look like is there's a housing area for ribosomes. Well, that could be. And they're showing the lysosome is right next to the ribosomes, which would make sense. So that could be. I didn't realize there was like a housing unit for ribosomes, but there could be. But let's see what happens. What does a ribosome do? I mean, what does a lysosome do with a ribosome? What's the purpose of a lysosome? Okay, here it is right here. The word lysosome is derived from the Greek words lyso, meaning to digest, digestive, and soma, meaning the body. The lysosome is a membrane-bound cell organelle. It's inside the cell and has its own membrane. It consists of the digestive enzyme. It consists of digestive enzymes, which digest the different intracellular and extracellular particles. They have to be broken down. They have to be broken down. If they're not, that's when you get toxic buildup and all kinds of other things. Now, lysosome consists of a variety of hydrolytic enzymes, a lot of them. Hydrolytic just means watery, which are active in acidic conditions. Okay, so they're watery, acidic enzymes. These enzymes break down the complex molecules into smaller particles. You have to break them down. Large proteins break down into amino acids, carbohydrates into simple sugars, lipids into fatty acids. These simplest particles act as nutrients to the cell. This is how you get you got to break the stuff down so you can absorb it. If these complex molecules can't be broken down into a simpler form, then it is called lysosomal storage disease. You can't break them down. Example, Hurler syndrome. In this condition, there is a defect in the metabolism of the mucopolysaccharides. Lysosomes destruct the different damaged cells by the apoptosis process, which means to destroy them, break them down, and then consume them and get them out of the body. That's cell apoptosis. And every cell finally gives out and has to be destroyed and removed. So the lysosomes destroy the different damaged cells. They're damaged, you got to get rid of them by this apoptosis process. Similarly, lysosomes destroy foreign particles like bacteria and prevent infections. Lysosomes and ribosomes, as far as I'm concerned now, look to be primarily your immune system. They're the things that keep invasion on that side. All right, what you have to understand is there's all kinds of bacterial different species. Some of them create things like this that alter our DNA. That gives us immunities. All right, so other ones don't have anything to do with our DNA. They break down chemistry, they break down fats and acids and dead things, and then other ones don't make ribosomes at all, I don't think. They come and they consume all that stuff. And then with their flagellas, they zip out through the highway and they dump that stuff off into the lymph area and into the lymph nodes to be processed to make it able to get out of the body. So there's a whole scenario of situations going on in here. It's not just one type of bacteria doing one type of thing. No, there's all kinds of different. Some of them invade DNA, which is the ones that make you sick or heal you or make you immune, and other ones feed you and clean up after you. You know, I mean, it's absolutely incredible. And the other thing is that I don't think anybody understands that. These, I believe, are the things that evolve. In other words, if they're going to make a ribosome that's going to make you immune from whatever it is, and it's never existed before, well, bacteria have to be the one to create it, or now they're doing it chemically. They're just snipping and clipping. They call it CRISPR. They can snip out little pieces and inject other pieces into DNA. So, you know, I'm going to stay out of that because that opens up a big door too. And that has nothing to do with me. I know this is all research. I'm just telling you how the human body works. What they do outside of that, that's not my realm. I don't get involved in that. But I can tell you what, these DNAs inside these bacteria have to change. They have to change to reprogram the ribosomes, to shoot out new types of ribosomes, to counteract the new diseases that pop up. There's no question whatsoever. These are the things that evolve. And they're going to have to, they must be very, very intelligent as far as I can determine. because this is programming. This is, and there's like 20 different amino acids. They can be programmed in all kinds of ways. So the complexity is just phenomenal. And DNA itself is yikes. And 98% of it is not junk DNA. That's where your memory and your immunities and your emotions and everything about you exists. And maybe even your soul and your spirit. I have no idea about that stuff right now. But I am looking into the chemistry of everything. Because everything there is chemistry. 100% is chemistry. There is nothing that happens that I know of that's not chemistry. And when I say chemistry, sunlight is, to me, is chemistry. All it is the smallest bits and pieces of chemistry. And that's my dipole electron flood theory. These are the two smallest particles that exist. It's a black and a white one. It's not shown perfectly in that picture, but it is online. Hold on a second. All right, I just want to leave one last word, and I'm going to finalize it at this. Everything there is in our bodies has to have catalysts to work. It's called nucleophilic substitution and invasion. It works with the half-lives and the isotopes, because all these isotopes have half-lives. But with the enzyme, it goes and breaks these half-lives down instantaneously, within nanoseconds. Done, which would take years without these enzymes. They're very, very exotic. All right, before we can even get anywhere, they're going to have to understand dipole electron flood theory. And protons and neutrons are not these big elementary particles that can be smashed into quarks and little tiny things like that, leptons and so forth. No, these are this. This is a proton. It's 1,823 dipoles. And what is a dipole? It's a positive and a negative. Or in the case of this, in atomic, subatomic, they can separate, and the black particle goes to the center. The glowy particles, the different colored ones, they stay around it on the outside, and the blue is the closest. All right? Now, so you never see the dark matter. They say, we look at everything. We looked at everything a million times. We never saw any dark matter. Well, they got to get inside, because that's where the dark matter is, right there. All right, but they never get inside, because they don't. The only way they get inside is with the super colliders, and when they do that, it's just such a mess they can't determine anything. Ours was just perfect. We could see these particles just like they were right in front of you. All right, this is their problem. They were using these huge things, smashing them, getting all kinds of debris. They can't make anything out of anything. But they're seeing the same particles that we're seeing, only they don't know where they came from. We know because we started with particles that were this small. We didn't start with these huge particles like this, so they just get debris. So what could we see? Well, this is what we could see. We could see the actual light interfering with other particles in the air, gases and so forth, and making them glow, and then accelerating because of the Venturi. And then the particle itself is actually seen, and here's the particle itself. And there it is. It's dipole electron flood theory. That's the actual particle. And when it comes through the Venturi out the other side, we see the Higgs fields. All right, I know it sounds crazy, but it's true. In order to understand any of biology or chemistry or anything, you have to start with a new atomic periodic table. Hydrogen is not like one gigantic proton and one tiny little electron, like one little tiny negative only particle and one big gigantic positive particle. They would just go together. There's never been any way they could, and I knew this years and years and years ago. It's called dipole electron flood theory. There has to be dipoles. It's made of dipoles. And there's 1,825 or so dipoles altogether. And it gets to a certain point and it's stable, and then it says, I don't want any more, but some more want to come in, and they float around outside, and they say, can I come in? They say, no, we have plenty. And I say, can I stay right there? They say, you stay there, you're all right. That's what's called a quantum distance. And the bigger this thing gets, the more want to get in. They love that black particle in the center. They want to be attached to that. That's what gives you all the energy. but there's no weight. The white has no weight. It's very, very, very strange. And here it is right here. That white has no weight whatsoever. Literally none. But there's a black particle attached to it, and these are the black particles. They have all the weight. And they don't get smaller or bigger. They're a fixed particle. And that's it. Fermilab has that right here on the Fermilab site. There it is right there. These are the two particles I just showed you. This one's fixed, and that one gets big and small. And it says that the extended particles, which is the black ones, have a fixed size, fuzzy edge around them. Yes, they do. The point-like particles have a zero size, they say, but they can't have a zero size. The zero size particles has extended effect because of the field. And yes, I agree with both of those statements, but it can't have a zero nothingness. But these are the things, and two of them back to back make up a photon. So when you get into dipole electron flood theory, wherever it is here, we have the same thing, only instead of this being a big positive, it's all dipoles. So we need to change the periodic table. Standard model's not right whatsoever. We got to change the periodic table. Electricity, gluons, heat, so forth, light. Chemistry's got to change. Space is just crazy. It's insane, the things they print about. Oh, we just can see to the farthest ends of the universe. They're still using the fact that light doesn't slow down. Of course it slows down. Redshift is 100% crazy, basically. The red shift, they say it's red because it's pulling away so fast because everything's expanding. That's the only way, because Einstein said it can't travel faster or slower than that certain speed. Well, that's insane, because I can show it speeding up and slowing down very, very simply. All right, I get these every day. We can see everything now with this Webb telescope and so forth. Well, first of all, they're looking through all of this stuff and saying light doesn't slow down. That's insanity, literally insanity. $10 billion cost for this telescope. And we're getting able to see these pictures. Well, that's nice. The pictures are nice. But that's a lot of money for some pictures. When we know that, you can see pretty good without this thing. What is that going to do for us? They're saying this is as far away as the cosmos gets. They have no clue, zero clue whatsoever. I would think we should stay focused on our region in space here and get ready for what we got coming, and it's coming. I can tell you that right now. It's not going to be pretty. All right, so we know they speed things up millions of times, and the bacteria create the ribosomes, so we're done with that. Now, what else? Better said, bacteria are the factories that produce the enzymes. The enzymes are what we need. Now, I'm going to show you how that does happen. So that takes care of that. And this is cancer invading. Stage 1 is here, stage 2, stage 3. Stage 4 is when it breaks through this new layer that they never even knew about. And it's called the interstitium. It's over here, right here. Meet your interstitium, a newfound organ. They thought this was flat and just a sheet. But it's not. It's a fluid-filled highway. And I've been investigating this for almost 15 years now. And I think I fully understand how this works in the immune system. And I'll show you that clearly. In membranes, this is so simplified, it's unbelievable. This is what they show for a phospholipid membrane. See, phospholipid. What's phospho? Phosphorus. And the reason I was studying this is because of aluminum silicates, which is feldspar. I could see all these things in my mud fossils, and nobody else was looking into them. So anyway, that's how I got studying this. And when these mud fossils were made, the phospho dropped down into aluminum silicates. This is a little bit complicated, but not bad. Remember I showed you all those little holes? These are protein channels. Right, they allow things to come through, and we'll have to talk about that in a minute. And these are just fluid-filled chambers. Now, I think there's another little layer on top of this that it isn't shown here, which is the fluid-filled highway. They always thought was flat. But you see all these glycoproteins with the carbohydrate attached. glycoprotein lipid with carbohydrate attached, cholesterol is floating around in there, integral membrane proteins. You know, they have a basic understanding of a membrane. This is very basic, very, very basic, because of the membrane itself is here, but they don't show the membrane being as sophisticated as it is. The membrane itself is extremely sophisticated. Hold on a second. I think I mentioned I found this in the mud fossils that I had discovered, petrified soft tissue. This is literally almost looks like it could be alive a couple of days ago. Here it is right here. All right, you got to put a little moisture on it, but that's it right there. And this is all marbled like meat. And that right there is the interstitium, is the fluid-filled highway. Now, there's two little dots I'm going to show you here on this little video I did. I'm going to be pointing down here at, and now this is up in my microscope. You see this? Those are those two little dots right there. They look like a guy's two eyeballs. Those are right at the tip of this little latch, and that's a latch that locks this into the next section of meat, whatever it was. And these, I believe, are the fluid-filled highways that transfer that fluid into the next organ or whatever it is. And it has to be some kind of a separation of chemistry, I would think. I really don't know, to be honest with you, but you are nothing more than a bag full of chemicals, as I am. We all are. And in order for life to work as fast as life has to work, you need the enzymes. To get the enzymes, you need the bacteria. And here's how the enzymes are created: by ribosomes, which are created by bacteria. You know how I showed you this here, this piece of meat with the two little spots on the end where the interstitium goes? Well, all of my stuff that I have basically preserved so fabulously that it's hard to believe, but it's true. Now, this is a bone, and this is what they call feldspar, all right, which is aluminum silicates. I agree with that. So I studied. I said, it's a bone. They said, oh, no, that's just aluminum silicates. That's just a feldspar. I said, well, it was a bone. Nope, and nobody would accept it. And inside was the, actually the marrow changed differently and turned into what you might call basalt. Now, there is one little spot of bone here, but it was just too much for anybody to accept, because it was such, you know, that's the only thing that would preserve was a bone. How could it turn into a mud fossil? Well, it was invaded, nucleophilic invasion. So I went and I started looking up the phosphorus, which is the membrane, is made out of phospholipids. Well, when those break down, their half-lives, they turn into aluminum silicates. See it? Aluminum silicates, aluminum silicates. They started out phosphorus of all different molecular types. Now, this is where they don't understand once again. These are all phosphorus, but they're not the same phosphorus. This one here contains not enough particles, and you add another one, and then add another one, add another one, add another one, and then it becomes stable at this number of particles, and it becomes stable. Now, some more want to get in there, so they come trying to get in from this side. Those turn into sulfur. You see, these are the half-lives and the daughter isotopes. Decay mode, the half-life, the da-da-da-da-da. How fast they turn into the half-life. And these are the enzymes that break these things down instantly. The phospholipids turn into aluminum silicates almost instantaneously. You saw how fast they work. So these, again, this is stuff they just don't understand it. You can't understand it until you understand dipole electron flood theory, because everything's made of dipoles. The standard model never works, still doesn't work. Gravity is the dark particle, the black particle, attracting the white particle. So here's the green is the same as the red. All right, here's the red right here. Exactly the same as I showed you from Fermilab. Only we see it because it's light. And here it is just accelerating and exploding, and we found this here, which is they've been looking for forever: muon neutrinos, electron neutrinos. The muon stays black, the black ball right there. It goes all by itself, called sterile muon, and the white squirts through here because it can, and it turns into showers. There it is right there, electron showers. That's fission right there, fission. That's fusion when they come back together. This was done long ago. And light spins in a circle. It's not a flappy wave like this. You see that? That's going through a slit. All right, that's like a drill bit going through there. And we can see them spinning. The red, this is the blue. The blue's the only one you can really see slowing down. It comes through really hot. And then you can tell there is two particles. The blue is a sizzler. The red and the green are not quite as fast. Here's the green coming through, and there's a green particle actually. It's the same particle. And these are the neutrino phases before they reach photon phase. And the red one does the same thing. All right, so before we can get to understand any of this, you got to understand the basis of what an atom's made out of. And then we can maybe understand some of this other stuff. Right now there's a lot of guesses. And I would like to see a bacterial database of the bacteria that's in the body. We know there's trillions of them, but how many different strains? And what strains affect what, tissues? Because every one of them is going to be a separate bacteria that protects a separate type of organ. Your kidneys aren't going to be the same as your heart. The membrane around it is going to have to deal with different chemistry. Your liver is not going to be the same as your gallbladder. It's all different types of chemistry provided by different type of bacteria, creating different ribosomes, which are the enzymes, which are the chemistry. You don't have the chemistry, you're not going to get that bazillion times a second interaction. And I mean, it's a fast interaction, and you can get sick real quick, and you can get better pretty quick too. It appears. I mean, there's spectacular remissions, and there's spectacular not remissions. So anyway, I'm going to leave it at that for today. And again, this is no advice given here whatsoever. This is research, and we do need some doctors to start looking into this and set up some studies or whatever, something. It seems crazy to let it just not be looked at as it should be. That's my feeling anyway.
Once you know the truth, there is no excuse. Once you know the truth, there is no excuse.